Okay, welcome back guys. I am Dr. Klim Raza. Today in this lecture we are going to discuss about the neural tube, the parts of the neural tube and different defects associated with the neural tube. Basically our neural tube is having three primary vesicles. So let me draw please. That neural tube is having three parts. So first part, second part and then third part. So three primary vesicles of the neural tube. First is the forebrain. This is called as prosencephalon. Prosencephalon. Then mesencephalon. And then we have got rhombencephalon. Rhombencephalon. So this is like forebrain. This is our midbrain. And this rhombencephalon is your hindbrain. Okay, these three primary vesicles of the neural tube are further subdivided into five secondary vesicles. So what are the basically parts of the prosencephalon? Prosencephalon is further subdivided into telencephalon and then diencephalon. So in the telencephalon we have got the cerebral hemispheres. We have got lateral ventricles also, which is major part of production of the CSF, and then we have got these other nuclei also important, basal ganglia nuclei also. So in the telencephalon we have got cerebral hemispheres, we have got lateral ventricles, and then we have got basal ganglia structures. And in diencephalon we have got thalamus, hypothalamus nuclei, and then we have got third ventricle. So it's mean that prosencephalon is further subdivided into telencephalon and then diencephalon. In the telencephalon, this is basically your structures present in your the cerebral hemispheres, lateral ventricles, and then we have got the basal ganglia. In diencephalon, we have got thalamic, subthalamic nuclei, and then we have got the third ventricle. So what are the structures in the mesencephalon? In the mesencephalon, if you remember that when we have discussed this, CSF, ventricular system of the brain or the subarachnoid space, and I have mentioned that, third ventricle is connected to the fourth ventricle with the help of small, aqueduct that is called as aqueduct of Sylvius or the cerebral aqueduct. So cerebral aqueduct is basically present in your midbrain in this mesencephalon. Then in the rhombencephalon, we have got two subdivisions like metencephalon and then myelencephalon. Metencephalon and then myelencephalon. So it's mean we are now at the level of the brain stem. At the level of the brain stem. So what are the derivatives in the metencephalon? We have got pons and then cerebellum. Pons and cerebellum. And in myelencephalon we have got medulla and its associated structures. So we have to understand that basically development of the nervous system, it usually start third week of the gestation, and this is responsibility of the ectodermal germ layer. And neural tube is having basically three primary vesicles: prosencephalon, mesencephalon, and then rhombencephalon. These three primary vesicles further subdivided into five secondary vesicles like telencephalon, diencephalon. We have got this mesencephalon as midbrain and aqueduct of Sylvius. Then we have got metencephalon, and then we have got myelencephalon. In telencephalon we have got these structures. In diencephalon, thalamic, subthalamic nuclei, and we have got third ventricle. And then in the metencephalon we have got pons and cerebellum, and in myelencephalon we have got medulla. Now there are some defects associated with the neural tube. Now these defects are basically known as closed and open neural tube defects. In closed neural tube defect, we have already talked about this spina bifida occulta, which is the most common neural tube defect, and it's associated with the boning defect at the lower end of your basically vertebral column, that there is boning defect but there is no displacement of the covering of the meninges or the covering of the neural tube. So this is only limited to the bony canal. So there is no displacement of the meninges or the neural tissue. So this is called as simply the spina bifida occulta, and this type of defect is called as closed neural tube defect. In open neural tube defect, we have got the meningeocele and then myelomeningeocele. That due to that bony defect, there will be displacement of simply meninges or the neural tissue also. So this is basic difference between closed and then open neural tube defect. Then there are some more defects relating to this part, and one of them is called as anencephaly. Now, first we have to discuss neural tube defects of the prosencephalon. Prosencephalon. So, if a baby is without forebrain prosencephalon, it's mean absence of the forebrain. This type of defect is called as anencephaly. Anencephaly. Absent forebrain. Absent forebrain. This may be due to some gene mutations. And another defect that is called as hollow prosencephaly. Hollow prosencephaly is basically what is hollow prosencephaly? We have to understand that hollow prosencephaly is basically due to the mutations in some genes like SHH genes. SHH genes, due to the mutation of SH, sonic hedgehog genes mutations. If there will be mutations in these genes, sonic H genes, then what will be the complications? So there will be no separation of the prosencephalon structures. There is no formation of right and then left hemispheres. So it means there is no separation, there is lack of separation of cerebral hemispheres. So only you have got one hemisphere. So one hemisphere, it means you are having one lateral ventricle. So it means you have got one hemisphere, only one cerebral hemisphere due to the defects in the SSH genes. So SSH gene mutation, single hemisphere you have got single lateral ventricle, and then only one lateral ventricle. So this type of defect is called as hollow prosencephaly. Another defect, normally you have got basically in your cortex you have got the gray matter. This is basically group of the cell bodies, because there are a lot of important somatosensory and motor areas, auditory areas also, speech areas also, and inside the brain you have got a lot of myelinated axons, and if there is basically no formation of these raised part of the brains which are called as gyri, or these depressed part sulci, so it means your brain will be flat brain. So such type of brain is called as defect is called as lesioncephaly. Lesioncephaly. So what is lesioncephaly? That in lesioncephaly the brain is the flat brain, smooth brain, that there is no basically gyri or the sulci, and this is due to the defect in the neural crest cell migrations. So you have to understand these basic defects, that anencephaly, no forebrain, absent prosencephalon. Hollow prosencephaly, that normally you have got two hemispheres which are connected by corpus callosum. So if there is no separation of these two hemispheres, it means you have got single hemisphere. This is called as hollow prosencephaly due to SHH genes mutations. Do remember this, please. And then lesioncephaly, it means this is a flat brain, smooth brain. There are no gyri and then sulci. Such type of defects are associated in later part of the age in many complications, consequences like hydrocephalus, Tau syndrome, and some other complications also. Now we have to discuss some defects of the posterior cranial fossa. Posterior cranial fossa, if you remember, if you have learned the bones of the skull, you know that in anterior cranial fossa you have got frontal lobe, in the middle cranial fossa you have got parietal temporal lobe, and then in posterior cranial fossa you have got entire occipital lobe, cerebellum, and then all the structures of the brainstem, and brainstem continues as spinal cord through the foramen magnum. So this is very important, some basic orientation of the skull bones also. You must have to recall the base of the skull in order to understand posterior cranial fossa malformations or the posterior cranial fossa defects. So in posterior cranial fossa defects, so you should write that posterior cranial fossa posterior cranial fossa malformations or the posterior cranial fossa defects. Normally, I have told you that in posterior cranial fossa, you have got entire brain stem, cerebellum, and then posterior, extreme posterior part of your brain, that is the occipital lobe. So, in posterior cranial fossa, number one, if there is, normally, what are the structures that are protruding through the foramen magnum? That is spinal cord, termination of the medulla, and medulla continues the spinal cord. Then there are two vertebral arteries passing through this foramen magnum, enter into your brainstem and give lot of branches, entire brainstem blood supply and cerebellum also. Then this spinal part of the accessory 11th grade nerve is also passing through the foramen magnum. But if there will be displacement of some other structures like cerebellar tonsils, so if there will be displacement of cerebellar tonsils, cerebellar, it means I am talking about the cerebellum. If there will be displacement of cerebellar tonsils through this foramen magnum, cerebellar tonsils displacement through foramen magnum. Normally, there is no displacement of cerebellar tonsils. Cerebellar tonsils are sitting in posterior cranial fossa. So, if there will be displacement, herniation, protrusion of these tonsils through that opening, it means this is posterior cranial fossa malformation. This type of defect is called as Chiari 1 defect. Chiari 1 defect. Chiari 1 defect. Chiari 1 defect. So, in Chiari 1 defect, you have got displacement of one structure through the foramen magnum, and if there is displacement of two structures, like with the cerebellar tonsils, you have got displacement of vermis of the cerebellum also, which is connecting. Vermis is basically connecting two hemispheres of the cerebellum. Suppose this my pen is the vermis and this is the cerebellar hemisphere right and this is left. So it means vermis is connecting my both cerebellar hemispheres. So if there is displacement of vermis displacement with the cerebellar tonsils through this foramen magnum, it means there is displacement, protrusion or the herniation of both structures. So this type of defect is called as Chiari 2 defect or Chiari type 2 defect. Now this is more severe. Why Chiari type 2 defect is more severe? You have to understand first the idea of the structure, that basically there in front of these all structures there is the fourth ventricle, there is central canal, there are some important openings that open into subarachnoid spaces. So it means such type of defects are going to block your CSF pathway. So there will be no communication in fourth ventricle to the central canal and to the subarachnoid space. So it means there will be increased CSF pressure and that may lead to hydrocephalus, increase ICP. So this type of the hydrocephalus is called as basically non-communicating hydrocephalus. So non-communicating hydrocephalus, it means it's associated with Chiari type 2 defect also. And then what else is there in the posterior cranial fossa? You must have heard about Dandy-Walker syndrome. Dandy-Walker syndrome. So Dandy-Walker syndrome, what is the concept behind this Dandy-Walker syndrome? As I have told you that cerebellum, which is posterior side of the brainstem, connected to entire brainstem to the peduncles. So cerebellar hemispheres are connected with the help of the vermis. So if there is absence of the vermis, agenesis, absent synthesis formation of the vermis, it means that vermis is basically helping formation of the roof of the fourth ventricle. So I will just give you a small description here. That suppose this is your brainstem and here you have got midbrain, this is your pons and this is medulla. So this cavity is fourth ventricle and here you have got basically cerebellum, and this part of cerebellum here, this part of cerebellum, this is vermis. Now this is fourth ventricle, So CSF is traveling, this is aqueduct of Sylvius from third ventricle to the fourth ventricle, like this is fourth ventricle. Now this is a vermis, So if there will be absence of the vermis, this is your sagittal view. So if there is absence of the vermis, it means there will be enlargement of which ventricle? It may lead to enlargement of fourth ventricle. So CSF, now this cystic enlargement of the fourth ventricle due to absence of the vermis, agenesis of vermis, this is also very dangerous. It may lead to blockage of this entire pathway here, and this also lead to hydrocephalus. This type of hydrocephalus is also called as non-communicating hydrocephalus. Let me show you some important radiological structures associated with our today's topic as well. Here you can see this is basically Dandy-Walker syndrome. First focus on this structure please. Here you can see this part is the cerebellum. Here is the enlargement of the fourth ventricle. So cystic enlargement of the fourth ventricle. Here you can see the CSF, this whitish part. Even it may lead to enlargement of third ventricle also, and also associated structure of the thalamus and hypothalamus. This is your basically pons, this is midbrain, and this is aqueduct of Sylvius. Here you can see this upper part is the straight sinus. So this cystic enlargement of fourth ventricle due to the absence of the vermis, this is called as Dandy-Walker syndrome, and Dandy-Walker syndrome is also very severe because it may lead to hydrocephalus, non-communicating hydrocephalus. And then on this structure you can see there is displacement of two structures like cerebellar tonsils and vermis. So this type of defect is called as Chiari type 2 defect. So I hope that you understood our today's topic, and in next topic we are going to discuss about another very important assignment, that is syringomyelia. Thank you for your consideration.